Okay, so it's just gone 6 o'clock here in the UK, so I guess we should get started. Welcome, everybody. Thank you so much for joining us today for our exciting event, Smallpox to COVID, a comparison of vaccine development. We have some fantastic speakers for you this evening, and we're so excited so many of you were able to join us. I'm Gail Chapman. I'm the Public Programmes Officer here at the Royal College of Physicians Museum. And joining me today are our fabulous speakers, most of whom you can see along the bottom. I'm just going to do some introductions in the order that they're going to be speaking this evening. So first up, we have our very own senior curator here at the Royal College of Physicians Museum, Larry Jones. She'll be followed by Anna Dhody, curator of the Mütter Museum and director of the Mütter Research Institute. Then we'll be hearing from Dr. Ana Duggan, adjunct assistant professor at McMaster University, who's also featured in our lovely photo. We'll also then be joined by Dr. Gregory Pollan, director of the Vaccine Research Group at the Mayo Clinic, who will be joining us shortly. He's not quite on screen yet. And we also have Professor Hendrik Poinar, from the Ancient DNA Centre at McMaster University, who you can see on screen. He will be joining us for the Q&A at the end. So you probably don't want to hear much more from me. I should hand over to our first speaker for the evening, which will be Larry Jones, the Senior Curator at the Royal College of Physicians Museum.
Hello everyone and welcome to our event this evening, Smallpox to COVID, a comparison of vaccine development. I'm really pleased that you've been able to join us this evening, and I'm also really pleased to welcome our sort of wide range of speakers that Gail has just introduced. I'm really excited to hear what everyone's got to say. So this event is part of the RCP, the Royal College of Physicians Museum's public programme, and our public programmes are generally linked to our exhibitions. So in my little bit to start with, I'm going to give you a brief introduction to the RCP, to the museum and to the exhibition, and how we came to be involved in the project that Anna and Anna are going to speak about. So for those of you who aren't familiar with the Royal College of Physicians of London, the RCP is a medical charity. It represents about 40,000 doctors worldwide, and its core mission is to improve patient health and reduce illness, so a fairly broad remit. As well as being a modern healthcare charity which works to raise standards and to support the medical workforce, it's also an organisation with over 500 years of history. So it was founded in 1518. And that history is looked after by the archive, library and museum team, which Gail and I are part of. And we care for a huge range of collections, so they're really varied, and you can see just a very small portion of them in this image that you're looking at. So our historic collections range from rare books to artwork, medical instruments, silverware, letters and manuscripts, and more recently recorded interviews with doctors. They cover the history of the RCP and the history of medicine, which are, as you can imagine, very similar but not quite the same. In more normal times, I think we're getting there, but still not quite there yet. So in normal times, these collections are all available for research, so they're all publicly accessible, even if they're not on display. But they are also displayed around our headquarters, which is in Regent's Park in London. And the collections are also obviously used in our active events and exhibitions programme. At the moment, we're waiting for a reopening date, but if you keep an eye out on our newsletter and our social media, you'll be the first people to hear when we do reopen. The activities of physicians over the years is our focus in the museum, and this is both their lives as individuals, so items such as their personal papers, as portraits, items they collected, but also their medical practice, so items that they used while actually being doctors. And one example of this I just want to quickly show you because it's quite topical and quite fitting for this evening. So this is something that we recently acquired from an individual donor, very kindly offered it to us. This is a smallpox diagnosis kit from the 1960s or 1970s in Britain. This would have been used to take samples from skin complaints that were suspected to be smallpox. The samples would have been taken by a GP, so a doctor in general practice, and then sent off for testing and identification to a central lab in London. This lab would then use the results. So if it was smallpox, they would use that for contact tracing to help prevent the spread of the disease. And that might sound quite familiar to some of the things that we're used to at the moment. This is essentially the 1960s version of test and trace. I mentioned that our event programme is linked to our exhibition, so I'll quickly tell you about our current exhibition, which is called RCP Unseen. Lots of museums have way more items than they can ever display at any one time, and we're no different. So we use lockdown and not being able to get in the building or to do a lot of the activities we normally do to really explore and have a think about our own collections and to pull out items and stories that we've not had a chance to show you before. We've put our exhibition online so you can see it on our website, but we will also hopefully be opening it physically at some point in the future. But as well as showing you kind of these slightly unseen, unheard stories, I also wanted to reflect what goes on behind the scenes in the museum, because I think there's this almost trope of museum and archive collections being dusty and quiet and unused, and that is really not true. So as well as reaching our audiences through various events and exhibitions, we also conserve collections to preserve them for the future. We actively collect so that we can tell today's stories again in the future. But our items are also used in a variety of research projects. This can be historical, scientific, medical, all sorts of things. And this is specifically talked about in the researching the collection part of RCP Unseen. And that is what you're looking at the moment. So there's, as I mentioned, there's a bunch of different examples of how our collections are being used for research. And I'm just going to quickly tell you a little bit about two of them. So the image in the top left reflects a PhD that's currently been done on our their book collection, which is looking at women's ownership of medical knowledge. So that is the book in the top left corner. Other research has been looking into the investigations of the work of Isaac Newton, so that's what that prism relates to. And then, importantly for this evening, this item. So this is a set of surgical blades. The handle is made of ivory, so it's just one handle and a set of interchangeable blades, all slightly different shapes and sizes. And you can see that the handle's inscribed with the name E. Jenner. So lots of the items in our collection were donated by doctors in the past. And, as you can imagine, in the past, the record keeping wasn't always that great. So we can't be certain about this, but it's believed that this set belonged to Edward Jenner, who you may have heard of because he's famous for carrying out the first controlled vaccination against smallpox in 1796. Jenner did actually have connections with the RCP, so the RCP promoted his work, which was not something it necessarily would have done at the time. And he was important in both his own time and, obviously important historically. And this would have meant that items related to him were probably quite collectible. They would have appealed to 19th and 20th century doctors as much as they would today. And these past doctors often collected things themselves, which is probably why we have it in the collection. So when we were approached in 2018 by McMaster University and the Mütter Research Institute to see if we had, any objects in our museum collection that could have been used in vaccination in the past, this was the one that jumped out to me. We can't know for definite if these blades were used to vaccinate people, but the attribution to Jenner made it seem likely enough that, we all agreed it would be worthwhile to investigate. And because of this, the set was then sampled in 2019 by Dr Ana Duggan as part of the A Pox in All Our Houses research project, which we'll hear about in a little bit. But firstly, I'm going to hand over to our other Anna Dhody, who is curator at the Mütter Museum and director of the Mütter Research Institute. I hope I've got your titles right. And she's going to tell you how the project came to be, kind of give us a bit of scene setting.
Thank you. Wonderful. So thank you very much. I'm very excited to be here. And what I'll do is I'll just give a very 30-second introduction into the Mütter Museum and the College of Physicians of Philadelphia. So kind of similar to the Royal College of Physicians in London, we are not a medical school. We don't grant medical degrees, but we have the name college in our title, which often causes confusion. But we are actually a professional medical society. And, you know, in the United States, it sounds kind of, you know, like fancy, like we're the oldest professional medical society in continuous operation in the United States. 1787, which now here in this audience doesn't really have as much oomph to it. But, you know, in the United States, it's pretty old. And that's because Philadelphia is the home, or was the home, of the first medical school and the first hospital. And so even today, we are what's called like a meds and eds. We are a biomedical hub. And as of the last time I did my research about a year ago, or pre-COVID, it was about 1 in every 6 medical students in the United States go through Philadelphia at some point during their medical training. And so that's pretty impressive numbers. And that's important to know because the College of Physicians, because we're a fellowship-based organization comprised of mainly physicians, when the Mütter Museum came about basically in 1859 and opened in 1863, the core of the collection was donations from physicians to the college, to the Mütter Museum. And that's important because it kind of speaks to the types of collections that we have. The time period is very much mid-19th century is kind of where the core of it is. And that all kind of goes to the origin story of how did a pox in all of our houses get its start, and what is it all about? And it can all be summed up with one sentence, which is, I left my museum director unsupervised. And that's important because we had a plan. We were actually all the museum staff at the time, all 5 of us, were supposed to go on a team building field trip to another medical museum about 3 hours away in Maryland. It's actually the National Museum of Health and Medicine, which is it's a medical museum, but it actually only focuses on military medicine. And so that's the core of their collection. But most of my staff hadn't been there before. We were going to go on a field trip. At the last minute, Robert, and that's the gentleman there on the left giving us that expression, decided to bail out, to not go, and because he said he had some work to do. We didn't know what that work was, so we went on our merry way. Turns out he just decided to go spelunking or cruising around in our mobile storage collection, which houses about 16,000 museum objects. Now, we're known mostly for our specimens, our biological objects, but as Lowry was saying, there's so much that museums have that's unseen. And so we have these amazing, predominantly in that room, instruments and diagnostic equipment, all these different things that date from pretty much the late 1700s, even up until about 1950. But again, the core is like mid-19th century. He was actually showing a new employee in a different department around and decided to open up our favorite drawer there, which is our phlebotomy drawer, our bloodletting drawer, because it has fleams and it has scarificators and it has all these things. And he starts to rummage around in there, and he had been doing some work with the Civil War, and specifically Civil War immunization. And so he was actually just primed to looking at this, and he realized, some of these phlebotomy kits don't look like phlebotomy kits. And he decides to open them up. I will say, thank goodness he was wearing gloves at the time. We did drill that into him. But he noticed that these kits didn't look like kits. So he takes them out and he opens them up. And sure enough, they were not phlebotomy kits. They were early, or I should say, they were vaccination kits that dated to the mid to mid-late 19th century. And they were comprised of basically, they looked like almost essentially, because the blood, you had to kind of use the same tools, like a lancet, for instance. So in many ways, they kind of did look like a phlebotomy kit. They had some of the same material, but that's where it differs, is that they also had these little compartments, little tin compartments, and in the middle picture, you can see what some of those compartments held. That is smallpox scab, sometimes referred to as matter. And then also, and I guess it might be hard for you to see in the upper kind of picture that's showing the entire kit, there's this broken piece of glass. And those were actually kind of glass slide plates that had dried lymph materials, like that clear fluid that oozes out of these pustules. That's also was in those. And that's because of the way that they propagated and the way they did the vaccinations prior to the much later in the 19th century, when they had more standardized ways of propagating the vaccine. Before that, what they would do, especially in the United States here, is person to person propagation. So you would have some matter that you got from another person who had been vaccinated, and then you would vaccinate that person. You had to wait a certain amount of time and then harvest the scabs and the lymph that came off that person, and then start the process over again. So these kits were very, very important in that process. And you can see they're very small, portable. They would go into the doctor's bag, and the doctors would go to the people to vaccinate. It was different. And so that was really important. And so what's really interesting, though, is that, you have to keep tracing it back, So where did that matter come from? Where it came from somebody before? It came from somebody before. It came from somebody before. And so we thought, hey, that's really interesting. But these things were going around in my head. But the first thing I had to do was I had to call my friends at the CDC. And that was a very interesting phone call. And in fact, it's never really a good sign when you call the CDC, leave a message, and they on their machine, they call you back in less than 10 minutes. Generally speaking, that's a public service announcement. Usually means you're in trouble. But they were very nice. And when I explained the situation to them, it was one of those things where they had to say, well, where are these kits now? And I had to tell the truth, which is that my boss put them on paper plates and they were in his office. So that was a fun conversation to have. Did I throw him under the bus a little bit? Yes. So he had to have a very uncomfortable conversation with 30 public health workers from the CDC and the Philadelphia Department of Public Health. And have we let him forget that? No, we will never let him forget that. But be that as it may, it went a lot better than it was expected. And what ultimately happened is, you can see kind of here, I'm in this lovely, flattering PPE. We had to take out all of the kits. We opened them all up. We documented them. We tried to ascertain how much matter and in what variety and forms we were talking about. But then ultimately, and this is kind of like the important thing, everything, including the kits, the matter, everything, went to our wonderful friends at the CDC. As is per protocol, that's fine. And the CDC had to do two things when they arrived in Atlanta. The first thing they wanted to do was to try to culture any of the biological material to make sure that it wasn't dangerous. And then so they did that, and they used a bit of the material to do that. And then they also wanted to do some genetic analysis on it. They wanted to kind of see, well, what are we talking about here? What types of orthopox would be used in this vaccination? Now, the CDC has state-of-the-art equipment. They are amazing. They've got wonderful labs. But, and this is where I'm going to transition it off to Anna, the other Anna, is that they're not an ancient DNA lab. And there's a very big difference between a modern DNA lab that works with fresh material and an ancient DNA lab. And again, Anna can talk a little bit more about that. But so what happened was, the CDC took it, in doing the necessary work that they did obliterate the vast majority of the biological material, those scabs and that lymph. And that was, it was a shame, because they did not get great results in their modern DNA lab. And so when I was talking to them, I threw out a question that I was pretty sure the answer was going to be no, which was, do you think it would be possible if we could take whatever material you have left over and the kits themselves, and maybe could we ship them across international lines to Canada? And to their credit, they didn't laugh at me, and they didn't say no right off the bat, because I explained that I had these great friends and colleagues at McMaster we had been working with for years on other projects related to cholera and all sorts of wonderful things, and that I knew that they had a great lab up there, and if they really wanted those DNA results, then these are the folks that we have to contact. And the last thing I'll say before I transfer it off to Anna is that since I knew that again, they were pretty they said we really used up most of the scabs and we used up most of that. I should mention that my background is in actually forensic science and forensic anthropology. So I was really keen on getting those kits up there because one of the things you learn in forensics is Locard's principle of transference. And that basically means anything that comes in contact with anything else or anybody else, a little bit of you or that object is left on that, and vice versa. So there's always a little bit left behind. And even if you can't see it. So I was curious to know, was there any viable material on those kits that they had used, especially the ones that didn't have any visible matter left in them? That doesn't mean that there wasn't anything there, but I did know one thing, which was going to be quite challenging. So I'm going to turn it over now to the other wonderful Anna to kind of continue this narrative on, their crazy friends down south and what they were getting you guys into. So over to you.
Thanks, Anna. So in this context, I'm the other Anna. I'm Anna Duggan. I'm a molecular anthropologist at the McMaster Ancient DNA Center. And what a molecular anthropologist means is essentially I study questions of anthropological interest, but I study them from the perspective of evolutionary genomics. So in this case, about really the practice of fighting smallpox as a disease, but also the study of the viruses that are associated with smallpox. So both variola virus, which causes smallpox, and then the virus strains that were used to vaccinate against smallpox. So I have to admit, I'm going to back up ever so slightly before picking up where Anna Dhody left with the kits at the CDC, and say that I had been studying smallpox for a couple of years before this, but it was a sort of an accidental finding. I had been working with a piece of tissue that had come from a mummy in Lithuania, and that was dated to the 17th century. And we had unexpectedly recovered fragments of variola virus, so the virus that causes smallpox, from that tissue sample. And I say unexpectedly just because that tissue had no sign of disease in it. It had no pock marks, no lesions, nothing to suggest that there might be something as significant as variola inside of it. And then the other piece of information that I guess I have to confess is that when that work began in, I believe that was 2015, I knew very little about smallpox, which I think is the privilege of my generation and the generations that have come after me, thanks to the generations that came before us, beginning perhaps with Edward Jenner, or even going further back to variolation if you wished, in that smallpox was really a historical footnote in my mind. I had no idea that it had this enormous disease burden, that it had mortality rates that could range up to 30% in some cases, and that it had really caused such massive mortality across the globe. The WHO states that their estimate is in the 20th century, when smallpox was really only circulating for 80 years, and that vaccination was pretty widely available, there was still approximately 300 million deaths attributed to smallpox, which is just such an amazingly large number that I had no concept of. And I think it's incredible that this disease that was human specific, that caused such enormous mortality, was eradicated and declared eradicated by the WHO in 1979, 2 years after the final naturally occurring case. And so in addition to my own edification about smallpox as it began with this Lithuanian mummy, the other interesting piece that came out of that project, again, circling back to my background as an evolutionary genomicist, is that with that 17th century variola genome, we were able to sort of reconsider all of the available 20th century variola genomes that are present in databases. So these are just sequences, right? They're letters that are in a database that correspond to what that genome would have originally been. And with this sort of anchor point in the 17th century, we can look at these 20th century genomes, which are all essentially samples that were collected during that final WHO vaccination campaign. They range from about the late 1940s to 1977. And we could see that there's these two clusters of 20th century variola virus. So one is variola major, which is the sort of version of variola virus that has that incredibly high, up to 30% mortality rate. And then the other sort of group of variola virus is variola minor, which had a much lower mortality rate, but also wasn't as widespread. It seemed to be more geographically restricted than variola major. And when we used our evolutionary biology principles with this 17th century Lithuanian strain as an anchor, we could estimate when those two groups of viruses split. And that estimate is, reasonably wide. It's measured in decades, not years. And the estimate that we came back with was really interesting in that it was in the end of the 18th century. So it sort of looked like it was possible that these two groups were sort of diverging into the variola major and the variola minor groups at around about the time that vaccination was perhaps becoming more widespread. And so that was sort of an idea that was in the back of our heads at McMaster, thinking, how can we study this when so many of our samples we find by chance, and we have just this variola, but wouldn't it be interesting if we could also think about what viruses were being used at that time point to vaccinate? And so what I think is interesting is, as Anna Dhody and Lowry have already talked about, is this practice of vaccination that began with this humanized vaccination, sometimes called Jennerian vaccination, where you would vaccinate one individual and collect that material and pass it on to the other, or onto another individual to be vaccinated. And one of the things that I think is so interesting and unfortunately still very timely today is that even though this practice was seen as being very beneficial, Jenner observed, other doctors observed, even large institutions such as the British military observed that this was a beneficial practice, that it prevented deaths, but they still weren't entirely sure what they were doing, right? This is still many, many years before germ theory. Interestingly enough, the word viral exists at this point, but we've never actually observed a virus in the 18th century. It's too small. Microscopes aren't strong enough to observe them yet. And so even though a lot of institutions had this idea that this practice was really beneficial, they didn't entirely understand what it was that was causing the beneficial outcomes, and weren't necessarily doing a great job at communicating that to the general public. I'm sure many of you have seen this Gilley cartoon. It's quite famous, and I've definitely seen it several times in COVID articles over the last year, on the BBC or various other news media using it. And what you see in this image, I'm not sure if you can see my pointer, but sort of in the center left, we have this very stoic, white-haired physician with his vaccination lancet, taking material from a vat that is labeled pox of the cow, and vaccinating individuals. And these individuals who have been vaccinated are starting to spout all of these cow-like protrusions. And so there was some hesitation even in the 18th and 19th century about vaccination. And I think one of the crucial takeaways from smallpox and still from COVID is that we as a scientific community, as a medical community, have never been really good at explaining to people why vaccination is important and why it is beneficial. And I think this is something that I hope events like this and that all of the communication that is happening in response to the COVID vaccine development can help. But this is not a modern phenomenon. This is something that has existed for a very long time. And so to go back now to where Anna Dhody left off with the kits at the CDC, how can you say no when a group that has a tagline, disturbingly informed, asks you if you want to work on their collection and they think it's really interesting and the CDC has signed off on it? I mean, of course, we jumped on the ability and the opportunity to work with them and to study these fascinating kits. And so, as Anna said, there were these kits that date to the mid 19th century. There were 5 kits in total that were sent. Some of them did contain biological or organic material like the scabs or the lymph. As Anna mentioned, some of that had already been used by the CDC when they did their checks. So the CDC really had a very different goal from this study than we did, right? The CDC was about protecting people, and they essentially said, there's no live variola virus. We don't have to be worried about setting off a smallpox outbreak in a population that now has multiple unvaccinated generations because vaccination of the previous generations was so successful. They were content that we weren't going to do any harm by studying this. And so given that there was limited amounts of this biologic organic material and not all of the kits had them, we started to think about this project from 2 different perspectives. So one, could we take the typical ancient DNA perspective and do what we call destructive analysis, which is like what the CDC did, where you take some of that biological material and you essentially dissolve it with enzymes to release all of the DNA that's held within, and it's gone, right? We can't put it back together afterwards. We've destroyed it, or we've consumed it. And then the other thought was, well, maybe we can start looking at destructive analysis, non-destructive analysis, sorry. Ancient DNA is focused on recovering very tiny amounts of damaged DNA. And so we started thinking, well, what if we looked at the objects themselves rather than just the biological material? And so we also sampled from the lancets that were included in the kits, the glass slides that would have had lymph between them, and also the tin boxes. And we were actually successful in extracting DNA from all of those sources. So this is really sort of opening a new world in terms of studying museum objects, in which we can sample things that don't have an obvious organic or biological residue on them, and we can do it without destroying or causing harm to the objects, which is great. As Anna mentioned, we in an ancient DNA lab operate a little bit differently. So first is that we're very concerned with cleanliness, as is the CDC. The CDC, again, has this opposite perspective, right? They don't want to cause harm to people. They're really worried about a sample making you sick. In an ancient DNA lab, we're not worried about that because, again, there's nothing infectious in our samples. So we're actually worried about getting our DNA in the samples and thereby contaminating it. So we wear the full Tyvek, we wear all of the gloves, the face masks, the hair masks. You can't see my feet, but I'm wearing disposable booties that are then in pairs of Crocs that we've bleached down the soles of those so we don't traipse around between rooms and spread anything that way. We're in a contained, filtered air system where everything is under positive pressure. So we're pushing air down and out so that dust isn't circulating and kind of hopping between samples or tubes. And we also, in addition to that, bleach and UV our workspaces a lot. And we also work in these what we call dead air hoods. So even though the lab itself will have this positive air pressure and will have controlled access where you can really only move between the rooms in one direction, you know, ancient DNA has to some extent been ready for COVID. We're used to following arrows on the floor and wearing masks for long periods of time. We also then keep open tubes in very restricted spaces so that they can be bleached and UV light filtered to destroy any DNA on a regular basis. And that DNA that we recover, as I said, is really damaged. There's no intact viruses here. There's no complete genomes. What we're usually talking about are very, very tiny pieces of DNA between about 35 and 50 base pairs long that, in addition to being short, have ends that have been damaged by being exposed to air and water and just no longer being inside of living cells. So, you know, I think you can kind of think of the ends of ancient DNA molecules, like your hair after a lockdown when you haven't had a haircut in 6 months and the ends are all split and fried and nothing looks good and fits together well. It's just, it's damaged and it needs to come off. And so from these tiny base pairs that are, you know, at best maybe 50 long, and we have genomes that are maybe 200,000 base pairs long, we have to sequence a whole lot of them so that we can use computers and sequencers to sort of computationally reconstruct what the virus was. So we're never putting the pieces of DNA back together to form a whole genome again. We're just trying to collect that information and then in a computer reconstruct what it would have looked like when it was living. And so we take two approaches. The first is shotgun sequencing, where we sort of, without bias, sequence any molecule extracted with equal probability. And that can be a really great first step to try and identify whether or not there's something of particular interest in a sample. So a virus in this case. And if we see that, it's probably present in very, very low percentage. And so then we can do this additional procedure called targeted enrichment, which is a little bit like going fishing for particular molecules. So you have these probes, which are sometimes actually called baits. And these are synthetic molecules that match your genome of interest. So you would sort of incubate your sample with these probes, and the hope is that your synthetic probe and a piece of DNA that's an approximate match will join, and then you can pull those out together. And the idea there is then when you resequence again after targeted enrichment, your molecules of interest that might have been present in a very, very tiny fraction beforehand are then present in a larger fraction. So you're getting more molecules of interest based on the total amount of sequencing you do, which is ultimately a financial decision in that it saves money in sequencing because you're not sequencing as much that you're not interested in, shall we say. And so from those Mütter kits with the shotgun sequencing, we identified two things that were really interesting out of the gate. So first, a really large proportion of the molecules we sequenced were human, meaning that when we compared those sort of 35 to 50 base pair reads against these large databases, the closest match was a human. And then the other was Vaccinia, which is a virus that we know was used for vaccination in the 20th century. And it becomes the predominant vaccine virus when vaccination becomes an industrial process. So with vaccine propagated in the bodies of cows or other animals, rather than this humanized vaccination that we associate with Jenner and the decades afterwards. And the human perspective, or the human proportion there, tells us that we had this virus that we associate with 20th century industrial vaccination, but used during humanized vaccination because this was material that was collected from a human body rather than from a cow. So this is, as Anna Dhody described, someone collecting either the scab or the lymph from within a pustule after vaccination to use it as the matter for future vaccinations. And because we had that perspective of the human reads, we could also understand a little bit about who these vaccinated individuals were who had their matter collected for future vaccinations. So using the mitochondrial genome, which is a small genome, we were able to identify them as having Western European ancestry. And based on the ratio of reads, again, these very short reads, that mapped as best matches to either the X or the Y chromosome in the human genome, we were also able to identify that we believe that at least 3 of those individuals were female. And so this is the sort of research that we're hoping to do with that E-Jenner blade kit that Lowry mentioned that we sampled in late 2019. Things have been slowed a little bit because of COVID restrictions, but similar to the process that we took with the Mütter kit, we did a non-destructive sampling of the blades and only the blades. We didn't touch the ivory handle. And so these blades would have been soaked in a solution with the hope that that solution would sort of release any molecules that might still be adhered to the outside of the blades into the solution that we can then extract and sequence them later. And we've got some preliminary sequencing results from these blades already. And from that, we can identify that there are viral molecules present. They do appear to be some form of orthopox virus, so that group of viruses that would contain not only variola virus that cause smallpox, but also many different viruses that we think may have been used for vaccination purposes, including vaccinia and cowpox. However, the number of molecules that we've recovered and the length of the molecules, so again, they're very short, hasn't been quite enough yet to allow us to really specifically identify what virus it is. And we're hoping that as we continue to work with these samples and maybe make some adjustments to our strategy in the lab, we might be able to identify that at some point, but we're not quite there yet. This is our hope, that we'll be able to trace back and start examining additional viruses that were used for vaccination in these time points. And so, taking a step back and again, the molecular anthropologist's sort of viewpoint from this, what I think is really interesting about these projects is the potential of them. And so thinking from an evolutionary perspective, you know, I still think that we're looking not only at the evolutionary history of smallpox as a disease, but the evolutionary history of the viruses that were used to vaccinate against it, as well as the virus that caused smallpox, but also the different practices of medicine, from this Jennerian humanized vaccination with the lymph and the scab matter to what eventually became like Lowry's more modern photo that she showed from the 1960s, like vials and needles, that how this practice changed over time and how different vaccinations changed with our knowledge that as it grew. And I think that information is held in museums and archives, and it's incredible. I think that perhaps we have before thought that information is held in paper records, in journals, in what physicians would have recorded about whom they were vaccinating, how they were vaccinating, what material they were using. But now we can take a 21st century approach to it and start to look at the actual matter that they were using that's left as things like residue on instruments, and recover that and reconstruct what those viruses were and compare them to all of the viruses that we have access to and know already, which I think is great. And I also just think that this project has only become more timely as we've worked on it, studying this practice of vaccination from really the birth of that as a practice with Jenner, and as it disseminated around the world from him to the global vaccination campaigns that we see now. Originally, I thought that this project was applicable to measles or polio, and then COVID hit. And I think it's incredible that it took about 200 years to eradicate smallpox via vaccination, and we had multiple COVID vaccines in less than 12 months. I think that's a really incredible achievement for humanity. And I hope that will work as a nice transition to Dr. Pollan now.
Thank you, Anna. Yes, that's wonderful. I think Dr. Pollan has joined us. So I'll just do a quick switch and put you up on screen. Thank you for joining us. This is Dr. Gregory Pollan, Director of the Vaccine Research Group at the Mayo Clinic, and many other titles on your slide there as well. And thank you so much for joining us.
My pleasure. Can you hear me well? Okay. And
yes,
you will
you
advance
great.
the slides, or I will?
If you would like to do it yourself, just hover over with your mouse and you will see there's a little back and forth arrow that you can click at the bottom.
Welcome, everybody. I was assigned the task of at least broadly talking about how vaccines are developed. So I'll describe vaccine types and the process of vaccine development, briefly describe the COVID-19 vaccine clinical trials, and then briefly discuss vaccine safety and hesitancy, all in 15 minutes. I'll do my best. I think an important point is that there are many different kinds of vaccines, and we can kind of lump them into two broad categories. Those that are passive, that is, we administer or provide the antibody that will gradually disappear, and I put some examples up there. And active, where we're giving something that stimulates the body to make longer acting antibody. And you'll notice that I have two in red font there, so-called genetic vaccines and viral vectored vaccines. Those are the two classes of COVID vaccine available in the UK and in the US, so I was going to concentrate on those. So let's look at the different vaccines that are available. I know this looks complicated, but bear with me. On the left-hand side, you see a variety of different vaccine platforms: a viral vector, a DNA vaccine, an mRNA vaccine. Then on the right-hand side at the top, you see that you inject that vaccine into the human body. Now I'm going to look just at the bottom. Many things happen, but several outcomes of interest happen. One is that you develop what are called T cells that are able to kill infected cells, that is, cells that are infected with the virus, so that we can clear the infection. The other major thing that happens is we develop antibodies, and I'll explain why those are important, and memory cells, so that after we get the vaccine, a year, 2 years, in the case of smallpox, 70 or more years later, your body remembers that viral pathogen and will quickly make antibody against it if it sees it again. Well, this is a cartoon of the coronavirus, and in particular, the SARS-CoV-2 structure. The woman that discovered this class of viruses, and she's not gotten the credit she's due, noticed under the microscope that the spike protein looked like a crown, and hence the Latin term corona. So this spike, which looks like this down below, is actually composed of 3 proteins. That is, it's trimeric. And so if you look to the far right-hand side, this is what that spike protein looks like. This is the outside tip of it. This acts like a key that goes around looking for the lock on our own cells that it can unlock and get into the cell, what you've heard as the ACE2 receptor. So what we do in a vaccine, in this case, is to make antibody against this S protein that coats this outside region so that it cannot insert itself into the host cell receptor and therefore cannot infect you. So we've seen 3 major types of coronaviruses in the West: protein-based, that's what the Novavax vaccine will be; viral vector, that's what the Astrazeneca and Johnson & Johnson vaccines are; and mRNA, the Pfizer and Moderna. I want you to note that they all lead to the same common final pathway. While they get there slightly different, the whole idea is to show your body what the spike protein looks like. And your body gets tricked into thinking this spike protein represents this. And so it makes antibody against that little portion, that little protein sticking out on the virus. So let's take the mRNA vaccines. All kinds of wild speculation and misinformation and disinformation about what these are. These vaccines started to get developed in 1960. In earnest, they started getting developed as vaccines in 1990. When you hear people saying they were rushed, that's like wearing a little sign that says, I don't really know anything about science, but I have an opinion. So these vaccines 30 years ago were being developed. This is simply a synthetic blueprint for the genes that produce that little spike protein. You put that into a lipid nanoparticle, it gets into our cells, the blueprint gets translated and makes this protein. Very simple, elegant idea, and it works. So the idea here is that if we can make this protein, we can produce an antibody against that protein. That antibody binds against the spike protein so that it cannot bind to the cell and cause infection. Lost my pointer. There we go. What mRNA vaccines cannot do, despite the misinformation you've heard, is it cannot enter into the nucleus. It cannot integrate into the host DNA. It cannot replicate. It cannot persist in the cell. It cannot mutate and code for something else. It simply is incapable of doing those things. So let's just take the first one, the Pfizer vaccine. It's the genetic blueprint for that one and only one protein, the S protein, takes 2 doses. The clinical trial was huge, almost 44,000 people. I'm going to show you some kind of sophisticated graphs, but this is what gets published in the medical literature. So if you look, the people who got, so they randomized, roughly 20,000 people got the Pfizer vaccine, 20,000 people got a placebo. They didn't know what they were getting, okay? And you watch and wait. And you notice that the people who got the vaccine, after about 10 days, no more people really got infected. The people who got placebo, they just kept increasing the numbers who got infected. So when you look at what was the ability of the vaccine to prevent infection 7 or more days after dose 2, it was 95% effective in doing that. Now, I want you to notice something. 95% is our best, what's called point estimate. The real answer lies somewhere between 89.8 and 97.6. So when you hear of, quote, breakthrough infections, those are people who perhaps were down here where 10, 11% of them genetically did not respond to the vaccine. No surprise, they would develop infection if they were exposed, or they developed a rather weak response, and while they got infected, had mild or even asymptomatic infection. All right, let's go to the Moderna vaccine. Same thing, the genetic blueprint for the full-length S protein, 2 doses, about 30,000 people. So 15,000 get placebo, 15,000 get the vaccine. Same thing. You see that the people who got the vaccine, they're not acquiring infection. The people who got placebo continue to develop infections. Let me just switch that for our modern day context. Here's what people who got the vaccine look like. Very rarely would anybody get infected. Here's what people who reject vaccines look like. That as a population, they will just continue to accrue infections, hospitalizations, and death. And again, our point estimate here was right at about 94% vaccine efficacy. When you break it down by various groups that you see here, you see that those point estimates vary from about 97.5% to as low as about 86.4%. And look at the point estimates. So it's people who are over the age of 65, the real vaccine efficacy is somewhere between 61.4 and 95.2. So don't be surprised that you'll hear of breakthrough infections. That is true for every vaccine ever invented and used. As Solomon said, there is nothing new under the sun here. What about the adenovirus vaccines? Well, I think for most of our listeners, which I believe are in the UK, you're familiar with the Astrazeneca, but there's also the Johnson & Johnson, Sputnik, and the CanSino vaccine. These work much the same way. In this case, you take the gene, the genome of a virus called the adenovirus. Adenoviruses are pretty simple viruses that cause the common cold, and you insert the gene that codes for the spike protein, inject it, it makes the DNA gets translated into mRNA, translated into spike protein, and the same common pathway. In the Astrazeneca, what they did is they took a common cold virus from a chimpanzee that encodes the same S protein, 2 doses. So if you look at the various trials, you'll see that if you got 2 full doses, it was 60% vaccine efficacy. If you got a low dose followed by a standard dose, it was 90%. This was very confusing. And it turns out that the real answer probably lies in the fact that if you got 2 standard doses separated by a longer time period, as is the general policy in the UK, you achieved very high vaccine efficacy. Less high if they were given close together to one another. Well, the way this happens is what we call phase 1, phase 2, and phase 3 studies. So phase 1 studies are designed to say, these are also called first-in-man studies. They're designed to answer the question, okay, this looks safe in animals, looks like it protects animals, let's try it in human volunteers. So people are brave enough, and really they're heroes in my mind, heroic enough to volunteer for these studies. We usually study about 20 to maybe 100 people to say, is the vaccine safe? Are there any side effects? Does it seem to work? If the answer to that is yes, we move to phase 2, where we study hundreds of volunteers. Again, looking at safety, and do we have the right dose and the right way of giving the vaccine? If the answer to that is yes, then we go to phase 3, where we have thousands or tens of thousands of people to answer the question, do the people who get the vaccine and people who don't get the vaccine, do they differ? Do they differ in side effects? Do they differ in efficacy? There's no way out of this. You have to do these studies, or we would never have a vaccine to protect against any disease. Like Dr. Duggan was talking about, if people had not done these studies with smallpox, you and me and our children would still be at risk for smallpox. Instead, we have vaccines that protect us because people entered these studies. So what does this look like as compared to today? Well, the typical vaccine development pathway I've listed down below here. It's usually 7, 10. The chickenpox vaccine took 20 years to develop, okay? That's at standard business pace. What do you do when the world's on fire, okay? When you have a pandemic, so right now, as of yesterday in the United States, 1 out of every 526 people are now dead of SARS-CoV-2. 1 out of every 526. Do you wait 10 years and do business as usual when it wouldn't be 1 out of 500? Maybe it would be 1 out of 100 would be dead. And instead, what they did is they figured out we could do some of the animal studies and the phase 1 studies simultaneously because we had data going back to 1990 when these mRNA vaccines were developed. And then they said, basically everybody put everything aside. We're going to, you saw in the Moderna trial, there were 99 centers around the world where they were enrolling people to get tens of thousands of volunteers and then follow them. So was this a faster development pathway? Absolutely. Were shortcuts taken? Administratively, yes. Safety-wise, no. And you've seen that based on the safety of the vaccines. So what are those safety issues? Well, anaphylaxis, about 2 to 5 cases per million doses, primarily in females. This is about the same rate as all vaccines. The blood clot risk you've heard about with adenoviruses. Well, it does depend upon age and gender, again, primarily seen in women, about 4 to 7 cases per million doses. Okay, remember what I told you. In the US, 1 out of every 526 dying. Which risk do you want? And this is something I think people don't understand. You don't get to choose no risk. You either get the vaccine and there's known risks, or you don't get the vaccine and you get infected with much higher levels of known risks. What about myopericarditis, primarily associated with the mRNA vaccines? About 20 to 30 per million, primarily younger males. What about what came out in the news a day or two ago, GBS or Guillain-Barré syndrome? About 8 per million doses, primarily in older men. Now, I gave you 1% here because people get scared. Oh, paralysis is associated with the vaccine. The answer is yes, in 0.0008% of people, i.e., a lower risk than getting hit by lightning. And yet we accept that, a much lower risk. I mean, if I showed you the risk of getting in a car and driving and lied to you and said that was the risk of a vaccine, you would never do it. So there's this spectrum we see in people from vaccine acceptance to hesitancy. And this is a review article I wrote in 2011 called The Age-Old Struggle Against the Anti-Vaccinationists. Every vaccine released in the US, including in the 1700s with smallpox vaccine, was met with equal degrees of skepticism, of rejection, and of hesitancy. And Anna already showed this, but I think it's a very pertinent one. We look at this and say, poor peasants, they actually thought pieces of cow would grow out of their arm. You tell me how that's different than people saying, oh, there are microchips, or there are magnetic microchips, or it's going to change my DNA structure. We will be genetically modified human beings. It's equally, let me be frank here. I don't want to be, I just want to be frank. It's equally as ignorant as the woodcut that we see from back then. Well, George Bernard Shaw said we learn from history that men never learn anything from history. And I think we've illustrated that. Full disclosure, my daughter pointed out he did say men. And then finally, George Santayana, well-known quote, many of you have heard, those who fail to learn the lessons of history are doomed to repeat them. And we are watching the same story play out between disease and vaccine as we have with every vaccine since the 1700s. And I will stop there. Thank you.
Thank you so much, Dr. Pollan. That was really fascinating. I'm going to put us all up on screen now. So just a reminder of everyone who's here. So we have Larry Jones, our senior curator from the Royal College of Physicians Museum, Anna Dhody, our curator from the Mütter Museum and Mütter Research Institute, Dr Ana Duggan, our assistant adjunct professor from the McMaster University, and we're also joined by Professor Hendrik Poinar from the Ancient DNA Centre at the McMaster University as well. If anyone would like to put a question in the chat, please do. We had one already that came in whilst we were presenting, so I'll kick off with that whilst you think and type. So the first one we had come in was directed for you, Anna Dhody. She wanted to know, Lindsay wanted to know, were you nervous when you came across this matter in the museum?
Oh, great question, Lindsay. Well, I mean, I have to admit my first reaction when I heard it, since I wasn't there, and I heard it via a voicemail on my cell phone in a van on the way back from Maryland, was strong language directed to my director, because, you know, of course, he said everything's under control. I put them in my office, and I'm like, So I'm thinking he's taking them out of the location, things, and all I'm thinking about is, my first reaction is, of course, he's making me call the CDC because he doesn't want to do it himself. And I wasn't damaged. I wasn't panicked. I switched gears to damage control. And so I wasn't even thinking at that point about cool applications of science. I was thinking about, how do we not get a giant, like, Christo tent put across the whole College of Physicians and just get shut down, because at that point, being shut down for any amount of time would be really bad. We've learned since that what happens when you get shut down. So we had to go through this protocol. So it wasn't until after all the material went off to the CDC and Robert cleared his pre-quarantine type thing where he had to keep reporting his temperature to 30 public health officials in Philadelphia and Atlanta for quite a while, that we could kind of step back and go, okay, everybody's okay, we're okay. And then we just kind of kept in touch with the CDC, and that's when we found out. I mean, again, it wasn't like we had an option. I mean, they had to go to the CDC, and they were really nice enough to communicate with us what they were doing. And they really didn't have to do that. They could just be like, okay, we're doing this, that's it, go away. But they were really nice. They explained everything to us, and they were asking other questions, and we had a good dialogue. And that's when I heard like what their desired outcome was. I realized that their ability to do that wasn't as great as McMaster's ability to do that. And so I was just in a fortunate position. If it hadn't been for another phone call that I received back in 2007 about intestinal specimens, I wouldn't have even known to contact Hendrik. So it's like this whole kind of series of events was kind of just odd and fortuitous. But I think it's it really boils down to like having these connections. These connections are key. Personal connections, these institutional connections, and being able to know and to place the right people and the right connections to do this type of work. And like, again, I really was really surprised and so grateful that the CDC was willing to do this. And I would say very grateful for Hendrik and McMaster because they had to go through a bit of hoops too to make it happen. It was not just the CDC. Then, as Anna Una was saying, it was the World Health Organization, and there was so much bureaucracy and hurdles and contingencies and everything. It was one of the big things I will say is it was a really amazing exercise in collaboration and communication. And for that, I'm just so thankful that everybody was willing to do that, because it's really hard.
Thank you. Yeah, it sounds, when you first told us that story, I was very impressed with how you dealt with it all. Okay, we've had a few more questions come in. So this one, I think, is probably Dr. Pollan. You would be best placed to answer this one if you wouldn't mind. We're wondering why some vaccines require boosters, which they're talking about doing in the UK.
Yeah. So when we give a vaccine, particularly against a new pathogen where we don't have any pre-existing immunity, you give what's called a priming dose. That's dose 1 of the vaccine, and then a booster dose, dose 2 of the vaccine. For some vaccines, we give multiple doses. For example, pneumococcal vaccine. For HPV or other vaccines, we might give 3 doses of vaccine. So it depends on the vaccine, depends on the pathogen, and it depends on how long you're wanting to stimulate immunity.
Okay, thank you. That's an interesting one for me because I've had the Astrazeneca and I've been told I'll probably need a booster later on at some point. So very interesting. So Maria's asked if, oh yes, just to say, Maria, we have been recording this evening and we will put the recording up on our YouTube channel, the Royal College of Physicians Museum YouTube channel, by the end of the month. So if you want to go back and remind yourself of anything you've heard this evening. So a couple of other questions have come in. Let's see if I can get through them all. So this one is for all panelists. Is there any benefit of your research to modern vaccine development? So maybe Anna Duggan, do you want to kick that one off?
Sure. Modern vaccine development. I think yes, though maybe not in the most obvious direction. You know, again, with an anthropological perspective, I think that a lot of what this research on smallpox and smallpox vaccines informs us about is the past and what was used. But I think, you know, as we live in a changing world, especially in a world with a rapidly changing climate, as we're seeing right now, you know, in Canada where I am, there's large portions of the country that are on fire. I understand that there is fatal flooding that's happened in the last 24 hours in multiple European countries. I think we're going to start experiencing more and more of these events wherein a changing climate introduces us to what are at least novel to our current population pathogens, but weren't necessarily pathogens that didn't exist in the past. So if we can look at historical diseases that were circulating in times of different human and nature interfaces, or in times of different sanitary or hygienic practices, or different climates that have now been frozen in permafrost or tundra and are about to be released. I think there's the application in that direction, though, perhaps not as direct as the COVID vaccine right now.
Thank you. Would anyone else like to talk to that
I
Okay.
might mention that we've learned some important things with COVID. That, you know, there's, even in the midst of tragedy, there are sometimes silver linings. One is that we've learned where we can move vaccine development forward very rapidly. We've learned how to use 2 new vaccine platforms. In fact, as we're speaking, there is a clinical trial now for an mRNA-based influenza vaccine. We're learning and watching in real time globally what happens when people don't take precautions and how viral mutations occur and spread worldwide. We're watching differences in the clinical consequences between the original SARS-CoV-2 virus, the UK variant, the India variant, the South African variant. These are essentially different viruses in many respects. And we're watching this in living color happening amidst a people and nations who reject science as a way of knowing. And so the consequences for humanity are severe and manifold. As I say to people, you know, this is a science-based problem for which we already have a science-based solution, and we can't come to agreement. How will we handle the much more difficult and complex issues of economics, of politics, of racism, of sexism, of global warming that Anna mentioned, et cetera? Those are far more complex than this. And yet, even with the support of science, People reject that as a way of knowing and believe they know, even absent any training in the field. That's a remarkable thing that was not, I don't think, as true in the past.
I would add one more thing is I know we've been talking a lot about virus too, but I think when we're talking about infectious disease, we can't leave out our friends the bacterium. And again, when I tell people that we're still in a cholera pandemic, we've had, I think, what is it, Hendrik, 8 or so, or 7 or 8 pandemics, and that in the time that we've had this, we're about an hour into, you know, this lecture right now, we've had like about over 30 people die of tuberculosis just right now in the world right now, and we're simultaneously dealing with these, and of course you're not seeing these types of infectious diseases in what, you know, we would call like the Western world, but they're still there. They're not going away, and we're also going to have to deal with antimicrobial resistance. So right before COVID hit, I was down in DC at a public health conference, and the two big things on everybody's minds pre-COVID were influenza and antimicrobial resistance. And they haven't gone away, and there's still issues. And one might argue that antimicrobial resistance is if nothing, maybe slightly exacerbated, but again, it still has not gone away. And so the work that we're doing with our collection, we've done multiple projects with historical cholera specimens. I'm hoping to be able to do more work with tuberculosis and other infectious diseases with Hendrik and Anna, because we need to know more about what they were doing to people in the past, how they were infecting people in the past, and how they have mutated and evolved as time has gone on. Because the more we know about the history of these pathogens, the more we will be armed with the knowledge to fight them in the present. And that could be said with all of these infectious diseases. And so again, the more we know about the past, the better informed we can be to be proactive, reactive, and just be better armed. So that's why these collections are important. That's why medical museums are important. That's why having that knowledge about what they are, what the potential of these, not just specimens now, but artifacts, non-biological objects is crucial. And I would say what is almost as equally crucial is getting that information out to the people who can then help us utilize it, and to help us get access to these things. And again, put these people and experts and institutions together who can do that important work. So that's where everybody who is attending this right now, you are now ambassadors of this information. And so I would take it upon all of you as a mission to spread this knowledge around. Maybe this is something that can go viral in a good way.
Wonderful. Thank you, everyone. We've had a couple of vaccine-specific ones for you, Dr. Pollan, if you wouldn't mind. So there's a question asking, do you think the speed of vaccine development and rollout is something that we'll be able to carry over into vaccines for other non-pandemic but still serious widespread diseases, maybe some of the ones that Anna has mentioned in the future?
Yeah, I certainly do think, and that was what I was alluding to, that we will take the lessons learned and figure out what we can safely do and what would not be safe to do. Recognizing that there are differences in expediency. As I say, if you were standing in a room and the room caught fire and you were engulfed in flames, if I said, well, I've got a new foam here that would suppress the fire, you wouldn't say, well, wait, how has that been studied? You do what you have to when the world's on fire. When it's not on fire and you're not having the surge in deaths, et cetera, you can, in a sense, afford to go slower, go age group by age group. You can afford to collect more data, whatever it is that would be of scientific importance. So you have to kind of put the public consequence into that mix of factors that determines what platforms we use and how fast we go with them.
Thank you. Yeah, I think that's an important thing. We also had a question asking quite specifically why it's relatively easy, you might argue against that, to generate vaccines against some viruses like COVID, whereas others such as herpes do not seem to be so easy.
Yeah, that's a good for our listener who came up with such a good question like that. And I wish we knew the answer. We have partial answers, but not good answers for everything. For example, some of the most difficult viruses to make vaccines against are those that are what we call hypervariable. HIV would be a good example of that. It's so variable that we can't make a stable vaccine against it. Let's take one that everybody's familiar with, influenza. That virus mutates so quickly that essentially every year we're changing one or more of the 4 components of modern day flu vaccines, hence the desire to make what are called universal flu vaccines. And again, it's different based on the peculiar properties of each individual type of virus.
Complicated. A challenge, definitely. Okay, oh, sorry, Hendrik, were you wanting to add something there?
Well, I was just gonna point out, so Jenner in a way lucked out.
Yeah, he did.
In the sense that smallpox was easily eradicable.
He not only lucked out, but he gets the credit because he was the first to publish.
An important one. Okay, I'm just going to switch around. We have a couple more vaccine questions, but I'm just going to go to one for Anna Dhody, or possibly Anna Duggan. In Anna's talk, you mentioned that some of the samples are identifiable as having come from someone female. Do we have any information about the gender of vaccine hosts in the past? Is that something someone's looking at?
Well, I think we can. It's actually in the terms of the studies that we've done and going back in the history. One of the things that was an issue wasn't entirely so much about gender, but about age. And so a big issue, especially during the Civil War, in vaccination and in using soldiers, generally younger men, and I'm not trying to be judgmental or anything here, was that they often were harboring other viruses in their body that caused what we call a spurious vaccination. Syphilis being one of the big ones, and when you had that, you had two things. You were having not very good smallpox, vaccination, but you were having great syphilis transmission among the platoon or the troop or whatever you were in. So one of the things they did figure out was that children were a better way to propagate this type of matter. So they would use children to actually get the matter and then take that matter and vaccinate the soldiers or the what have you, and then but not use the soldiers to then propagate it. So they had to go to a different source. So there may be other studies there about how sex was, if that was a factor involved. But in the minimal research and I should stress it's minimal, that we've done so far, because there's so much left to do. One of the things we did notice, it definitely was age. So children were better.
Okay. Thank you. So we've got a few more questions in. We might not get to all of them by past unless everyone's got a few more minutes. Another couple about vaccines specifically, if you don't mind, Dr. Pollan, someone's asked, this I think is probably a US question, why the FDA is holding out on full vaccine approval?
Yeah, it's not a holding out process, it's the administrative process. So for something to get a full license, FDA will literally go through every single one of the stack of records generated for each of those 44,000 participants. They will verify the laboratories where any assays were done. They will rerun every statistical analysis. They will comb through that data in order to be certain of what the safety and efficacy of those vaccines are. That is prescribed by law. So they're doing the right thing. They did the right thing, in my opinion, by releasing it under emergency use authorization. They're doing the right thing by taking all the prescribed steps for what's called a BLA, a biologics license application that would be approved. And I expect that we will see the first BLAs being granted probably in the September, October timeframe.
Thank you. And then there was another one about, so you've mentioned about obviously the percentages of the effectiveness of the vaccine. So Kim's wondered, is there anything that can be done to decrease vaccine resistance?
You mean vaccine hesitancy among people?
I'm not sure. I took it to mean, sorry, Kim, if you wouldn't mind clarifying in the chat, whether that's hesitancy or is it resistance to
viral mutants?
Yeah.
Yeah, well, let me answer both of them then. Yes, there are some things that can be done to decrease vaccine hesitancy, but it gets to an irreducible level, as it has with every single vaccine throughout history. Okay, so you'll never get everybody to agree that smoking is a bad thing and we shouldn't smoke. Won't happen. You won't get everybody to agree that, yeah, you should wear seatbelts, and you won't get everybody to agree that you should take a life-saving vaccine. In terms of actual viral resistance, the people who are unvaccinated at this point essentially are viral variant factories. So as they get infected, it gives an opportunity for the virus to mutate. That's why we keep seeing worse and worse viral variants. And then a number of those will have increased genetic fitness. That is, they will transmit more. So when you look at the original virus, whatever that transmissibility was or infectiousness, the so-called UK variant was 50% more infectious than the original virus. The Delta variant, or so-called India variant, is 50 to 60% more infectious than the Alpha UK variant. They're getting worse, not better. And it's happening because people are unimmunized. And eventually you could get to the point where the virus would be different enough that it could evade vaccine-induced immunity in part or in whole. So you will notice the vaccine companies announcing that they are embarking on development of variant-specific boosters against this. So, the cat's out of the bag. We had an opportunity to do this right. Populations and governments rejected that way of doing it. The consequence is, a pandemic that has killed well over 4 million people who would not have died otherwise, and a pandemic that will continue on through 2022 and perhaps beyond, and likely evolve into a virus like influenza that will now always be with us.
I could add one thing to that too, which is right about 4 months before the pandemic hit in the United States, we had put up an exhibition called Spit Spreads Death, where we examined the 1918-1919 influenza pandemic. And what we put in the exhibition and what we talked about, people, anti-maskers, issues like that. I should also say there was no vaccine available at that time, and there wasn't for decades subsequent, but just the other types of behavior that we mentioned in the populations and refusal to accept, certain bits of information and certain things were, again, the same quote as Dr. Pollan's last slide about history repeating itself. But I will say it was a particularly disturbing, as somebody who had just literally wrote that, been part of that exhibition, to see it happening in real time. And we were just saying, okay, well, we can point when that happened 101 years ago, and we're still seeing it happening now. I will say one. I mean,
I'm sorry, Anna, can I just interrupt? I'm
please,
so
yeah,
sorry.
yeah.
I'm 4 minutes late for a 1:30 call that I had, so I'm going to have to sign off, but it was a privilege to work and talk with you all.
Thank you.
Thank
so
you.
much.
Yes, I was just
Thank you
thinking
very much.
we're getting a little bit over. Thank you so much, everybody. Okay, so Anna and Hannah, who are still here, thank you for staying a little bit longer, and for everyone who's still joining us. Those questions that we haven't got to, we'll see if we can maybe circulate, and if we've got any further responses, we'll see if we can add those after the fact. But thank you everyone for such interest and for so many questions, and for all our lovely speakers who Professor Poyner unfortunately also had another meeting middle of the day over there, so many things to go to. So just before we go, all I wanted to do was say thank you for joining us this evening, and well, or this afternoon, and hope to see you all again at a future event.
Yeah,
Thanks
thank you
very
so much.
Bye.
Thanks so much. Bye.